All right, I've got gonna pick up where we left off. That'll work because I'm right in the middle. So I know I have a sub today out at the uh, SJS Division 3 sections with the track team as they're going to be competing to try to move on further on in the season. So we'll want to finish up these notes. This is where we left off. I had just been talking about the shape of DNA strand and wanted to let's see, move to the next one here, let you add on to those notes. We talked about the nitrogenous bases. Remember GCAT, I put them in different order, but it's adenine, guanine, cytosine, thymine. Those are the nitrogenous bases. There's a 5-carbon sugar called deoxyribose. That's where the deoxyribonucleic acid comes from, that deoxyribose sugar. And then we really look at the shape here. So you've got, it's double-stranded, it's double helix. I had just talked about kind of like a twisted ladder. It's a little offset. You can see it's uneven, but that's what gives us like these major groove and minor grooves and the differences in the way that it kind of like waves back and forth. But it is made up of like what we would refer to as the rails. Like if we could untwist it as a ladder, the rails would be the phosphate and the sugar, and then the nitrogenous bases, they come together and they bond. Think kind of like a zipper, but like they meet up, and those would be the rungs of the ladder. And so DNA is used in our cells to create proteins. And we talked a lot yesterday, kind of reviewed all of the different macromolecules and the function really of proteins. Well, there's a vast number of functions for proteins, but really like you kind of look at these, there's a lot of structural, and then there's a lot of communication components for proteins. So proteins are, most of what your body's made of. It's really important that these proteins are made the same way every time. They are a very specific set of components that have very defined roles in the body. And so it is important that the plans are always the same, and that they're consistently the same and consistently producing the same protein. That comes from the DNA. So DNA is the blueprint for building proteins. A code is the actual like order of the nitrogenous bases. It gives the like physical information for how a protein is to be built. And we're going to walk through the steps of that. And what I want you to remember is that we start symbolizing the nitrogenous bases as the letters, and you can kind of see that in this picture. C, G, A, A, C, 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 T, T, A, 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 A. But each of those letters is actually a representation of a real nitrogenous base, and that base comes in a set of 3, and they have those 3 codon and anticodon fit together, and they have with it, attached to it, a particular amino acid. And when your body needs a particular protein, there is a series of steps by which the DNA is used to create the protein. Okay, I skipped a step in there, and I'll talk about it in the bit to come. But that they're sectioned off per like the protein that needs to be built. So there's a start and an end, kind of like a paragraph. Okay, like a paragraph is going to communicate a complete thought, and it starts and it has a kind of a symbolic start. Sometimes we talk about that little indentation, or you can kind of see where that there's like a space above, and then there's a start point, and then the paragraph ends typically with some kind of a period or a question or exclamation point, but that it's defined and it is a chunk of information. Well, a gene is a chunk of information that is used to create a particular protein. And so this section of DNA, it codes for a protein. So what does a gene do? Codes for a protein. Okay. You have probably seen this before. We talked about cell division, looking at how cells replicate. This is indicative of how DNA is copied and distributed so that there's the same message going from cell to cell. And DNA is found in the nucleus in eukaryotic cells. So it's a little schematic, but this is a drawing that represents a pretty accurate picture of kind of the general parts of an animal cell. And the big area in the middle there, whoops, is showing DNA. Let me see. Can I get the little tools on here? No. Okay. Fine. And not prokaryotic cells. Remember that we talked about different types of cells, eukaryotic and prokaryotic. Prokaryotic cells don't have a nucleus, like bacteria. So, and let's see. So far, when you check your notes, you should be able to answer, or your notes should include, what are the parts of a nucleotide, what nitrogenous bases, and what sugar does DNA contain? How would you describe DNA's shape? And then what is the section of DNA that codes for a protein? What's that called? And where is DNA kept in the eukaryotic cell? Where is it housed? And it stays there, okay? The nucleus or the nuclear membrane only breaks down when the cell is like made a copy of all the DNA, organized them into chromosomes, and it's ready to move and to create two new cells with the exact same genetic information. All right. So now we're on to this new idea, DNA replication. What happens before a cell actually goes into cell division is that the DNA has to be copied. The code that, and you can see all these like little letters, they represent the nitrogenous bases, and each nitrogenous base only fits with another nitrogenous base, and the order in which they are arranged on the DNA is unique to the organism that houses that DNA. There are three major processes where DNA, RNA, and proteins are concerned, and the first is DNA replication. This is how DNA copies itself. The next slides show the steps of DNA replication, so you can actually kind of draw, and I'll talk you through a little bit of it. But it's really important that this is accurately done, because this information is going to be passed to the next generation, or maybe it's repairing something in your body, and that those cells need to function the same way that the cells did before. So it's got to be an accurate replication. If it's not accurate, if there are mutations, that can cause changes. Okay, so step one. Whoa, hang on. I think I'm going to use the arrows. Step one, the DNA has to unwind. There is an enzyme that actually facilitates this called helicase. It breaks down the hydrogen bonds of the nucleotide bases and unzips the DNA, basically. So you can see there helicase is circled. And then we have step two. It needs to be held open because there's an attraction between those bases, and it wants to just like reattach. And so it has to be held open, and binding proteins hold that space open. And then step three, there are new bases that are going to be added on to the DNA. So think like you're building, I don't know an analogy. Like if you're in order, something's coming in order, like kind of like beads on a necklace, but only if they match something that's already there. Okay. And the enzyme that is responsible for attaching the correct bases and matching those bases up, and then what you can see here is that one DNA strand gets unzipped and becomes, it's like half old, half new, half old, half new, but it's replicated because the bases are what's added. So this, the enzyme in charge of that is called DNA polymerase 3, and it begins laying down what we call complementary bases. So A and A would match, right? But nitrogenous bases bond in a particular way. So A goes with T. So a complementary relationship there, and the bonding relationship is that adenine and thymine would bond together, and that's what we refer to as complementary bases. And so there's a little primer chip, this little piece of RNA that attaches, And think of it kind of like the beginning of a zipper, the little thing that you have to zip, like get into the little, I don't know. Let's see. I have a zipper right here. So you got this little piece where you have to zip that and get it into the mechanism, and I feel like that's like the RNA primer. And then like the DNA polymerase is kind of like the zipper, if you will, and it does all of the work. But instead of just like a strand that's already there, it's like building a new strand as it goes. All right, so here are the rules of base pairing. We call these the DNA base pairing rules. I already said one of them in the adenine and thymine bond together, and they're held by what are hydrogen bonds. These are not covalent. They're a little bit weaker, so the unzipping and the zipping happens often. Hydrogen bonds hold the A and the T together, and you can see that's a double bond, and then the G and the C are bonded together, and that's a triple bond. And those are the complementary pairs. You can also see here in this illustration how the ladder is, like the sides of the ladder are structured. So sugar and phosphate, and how they are bonded as well. All right. Let me go back, make sure we got key features of DNA structure, partial chemical structure. All right. So take some time, pause, get this written in your notes. All right, step 4 is link fragmentation. So it's actually going to like hang on. Okay, so DNA doesn't just build all willy-nilly. It has to happen in a particular direction. And so when DNA unzips, there are particular markers on that DNA that are built into the genetic code that give a signal to the protein for that protein to bind there. And DNA ligase links what are referred to as Okazaki fragments on the lagging end strand, and replication is only going to occur in one direction. It only occurs for what we refer to as from 5' to 3' direction. And so when what you see these little arrows on the diagram right below my face, okay? So if I could joke about it, hang on. Right down there, okay? That on one end of the oval, it's going one direction. Hang on, not that way. There we go. I have to do it backwards. It's going this direction. And on the bottom, it's going this direction. I can't do it in the camera. So it's going in two opposite directions. And that's how fragments, they only are going to be joined in that one direction. All right. You can see that, like I said, replication doesn't just start on one end. It kind of starts in the middle of things. And it happens like in a bunch of different places because DNA is extremely long. And once these enzymes are introduced into the nucleus and replication begins, they are little workers. Like these reactions are going to happen when that protein is present. And so it happens continuously throughout the strand at replication bubbles. And you can see that this is actually a picture of DNA being replicated. And this happens until all the DNA in the molecule has been replicated. All right, so we're going to practice a little bit. And you can see that these are kind of set up in threes, and that's coming. We're going to talk about why it happens in threes. We're just starting with replication. This would not be a very long strand, but we are really just going to look at what would be the complementary side of this little DNA fragment. And so you have adenine, cytosine, guanine, cytosine, thymine, continues that way. So we'll read from right to left. And so I want you to pause, and we're going to write, let me see. Oh, it gives you the whole thing. Okay. We're going to write below the strand. Okay. So pause it and give it a try. A goes with, hang on. I'm trying to find the little tools that go with this. Because I should have like the little tool set that I could write directly on the screen, and I'm just not finding it. So darn. All right. Anyhow, so A with T, A with T, look over there, right? C with G, G with C, C with G, T with A. And so they have complementary pairings, and you're going to like write them as a sentence right below the strand. And each one needs to be lined up specifically with its base pair above it. You don't want things to get moved off from right or left. So check your work and see if you were correct. Okay, this is all over the place. I just like push one little bit and it goes. Okay, so we're almost at the end of the notes. So now on your notes, you should be able to find information about what the steps of DNA replication are and what the base pairing rules are. So for DNA, you can check A pairs with, oh, good job, thymine or T, right? And guanine pairs with, oh yeah, cytosine, right? C, good job. Okay. All right. So, like I said, it's always best. Get that Cornell set up. Got your questions on one left-hand column. Make sure that you can find the answers for those in your notes. Go back and add things to your notes. Write a summary. So make sure you refine those questions. Got a lot of them here, right? Let's see. Oh, I went past this. Whoopsie. Checkpoint. So there's some questions to add. And there are some questions that you can add. So within your left-hand column, you should put those questions. And then go ahead and write a summary at the bottom. Like, what's the purpose of these notes? What are we really answering? Let's go back, way back to the beginning. Okay. What are the components of a nucleic acid? And what are some examples of nucleic acids? And then we talked about what is DNA replication. So, and what's the purpose of DNA replication? That's a good thing to really understand as well. All right, the end result is another cell, right? 2 new cells from the one. Not 3 cells total, but 2. Cell division, man. All right, I think this is it. Have a good one.